Thank you everyone for the opportunity to talk to you today about some of the work that we've been doing looking at the crosstalk that happens between vasculature and neurogenesis during aging. So over the last 10 or 12 years, there's been a series of studies that have shown that there are factors circulating in the blood of young animals that can reverse some of the functional decline that happens in the tissues of old animals. And vice versa, there's factors in old blood that can be harmful to young tissues. And there's been two major experimental strategies that have been taken to show this. One is called parabiosis, where two animals are surgically joined to each other. Their vasculature merges with each other and they end up sharing a single circulation. You can either do this with two animals that are the same age, old, or young, and that's called isochronic parabiosis. Or you can join an aged animal to a young animal in heterochronic parabiosis. So in this condition, the aged animal ends up being exposed to everything that's circulating in the blood of the young animal and vice versa. And then in the other strategy, we can collect plasma from young animals or aged animals or from human donors, inject it into your experimental subject, again with the point being to expose an aged animal to all the protein factors that are circulating in young. Now, there's now been several studies from several labs showing that when you do interventions like this, you can actually reverse a lot of aging phenotypes in several different tissues. So, for example, skeletal muscle and liver both have the capacity to regenerate in response to some sort of insult or injury. That regenerative capacity declines during aging, but under conditions like parabiosis, it can actually be restored. So that means there's something circulating in the blood of young animals that can restore that regenerative capacity, and it means that the loss of regenerative capacity is actually reversible, at least to some extent. So in our lab, we've focused on what happens to neurogenesis in the brain during aging. So in the adult, there's two zones where neurogenesis continues throughout life. One is the subventricular zone, where neural stem cells reside along the wall of the lateral ventricle. They can proliferate, differentiate, migrate across this rostral migratory stream, and then newly born neurons integrate into the circuitry of the olfactory bulb. And then in the hippocampus, neural stem cells reside in the subgranular zone. And again, they can proliferate, differentiate, and then new neurons integrate locally into the hippocampal circuitry. So this process continues throughout life, and it declines as you age. During aging, you end up losing the numbers of neural stem cells, and they lose their ability to give rise to new neurons. So we asked whether doing something like heterochronic parabiosis could reverse that loss of neurogenic potential. So we generated either isochronic or heterochronic parabiotic pairs, and then looked in the subventricular zone for proliferative cells, here labeled with Ki67. We also looked at BRDU incorporation, or we looked at the numbers of neural stem cells labeled in SOX2. And what we see is that in the heterochronic parabiont, there's a significant increase in both the total numbers of proliferating cells and in SOX2 positive stem cells. And then if we treat the animals with BRDU and then look in the olfactory bulb, we can see that in addition to this increase in neural stem cells in the SVZ, in the olfactory bulb, there's actually an increase in the numbers of newly born neurons that have actually integrated there. And if we test these animals' behavior, there's improvements in their olfactory discrimination behaviors. And around the same time, Saul Vieta and Tony Viscore at Stanford have published a series of really great papers looking at similar effects in the hippocampus, where during heterochronic parabiosis or after plasma administration, there's an increase in the neurogenic potential in the hippocampus, and that's accompanied with improvements in cognitive behaviors. So in addition to these changes in neurogenesis, we also saw changes in the vasculature of the SVZ. So what you're looking at here is brain capillaries that are stained with the endothelial cell marker CD31. And on the left, this is the vasculature in the SVZ of an isochronic young animal, so a young animal paired to another young animal. On the right is isochronic old, so two old paired together. And you can see there's an obvious degeneration and fragmentation of the vasculature. But following heterochronic parabiosis, somehow that vasculature is restored. So the blood vessel volume, area, number of branch points is all restored in the old animal after being exposed to the young circulation. And to us, this is really exciting for several reasons. This being seeing the vasculature and neurogenesis change together is really exciting for a number of reasons. One is that blood vessels, neurons, and glia are all tightly coupled together in a neurovascular unit, and cerebral blood flow and brain activity are very tightly co-regulated and dynamically regulated. So for example, in fMRI, where we're imaging brain activity, what we're actually looking at is the consumption of oxygen in the brain and changes in local blood flow. And in the brain, as you age, blood vessels degenerate and constrict the same way that they do throughout the body, and this can be associated with a number of pathological conditions. The most obvious one is stroke, but it turns out that impairments in blood flow to the brain can have severe effects on cognitive abilities. So there's actually something called vascular dementia, which is the second most common form of dementia after Alzheimer's. And in addition to that, in a lot of diseases like Alzheimer's, which we think of as being a neurodegenerative disease, there's actually a vascular degeneration component as well. And then finally, and probably most relevant to this session, the vasculature makes up a key component of the neurogenic niches. So for example, in the SVZ, the neural stem cells are in direct contact with the CSF inside of the ventricle, but they also make contact with the cells that make up the blood vessels themselves. And so the circulation is providing both metabolic support to the brain, but also trophic support to neurogenic niches, and that can be either things that are circulating in the blood itself or the cells of the vasculature themselves secreting things into the CNS. So what can account for this phenomenon during parabiosis, where an animal is exposed to everything in the blood, there's some rejuvenating effect? Are there any single factors we can identify that can account for some or all of that? And a few have been identified. So this is just a list of a few: Klotho, beta-2 microglobulin, IL-33, CCL11. I should add TIMP2 to this list. And in our lab, we've focused on a member of the TGF-beta family called GDF11. So we sought to test whether injection of this single protein could recapitulate any of the aspects of parabiosis. So we injected recombinant protein, recombinant GDF11, into aged animals every day for 28 days. And then we looked to see what happens in the vasculature of both the SVZ and the SGZ in the hippocampus. And we see in both places an increase in vasculature measured by volume, area, number of blood vessels, number of branch points. And along with that, we see increases in both zones in the number of proliferating cells labeled by either Ki67 or BRDU, the number of neural stem cells labeled by SOX2, and the number of newly born neurons labeled by double Corton. And I should say here that we actually don't think, when we inject the recombinant protein, we don't think it's getting across the blood-brain barrier. So we think when we see things like this happening, that's actually some secondary effect that's happening from either the endothelial cells secreting something into the CNS or some secondary effect of an increase in blood flow, but we don't think it's coming from a direct stimulation of the cells by the recombinant protein. And then beyond just looking at the structural changes that we see in the vasculature or these changes in neurogenesis, we also think we're starting to see changes in actual functional changes in the brains. So, of course, red on black is not showing up great, but we can take the hippocampi from aged animals that have been injected with GDF11 and stain them for a marker of neural activity called delta FOSB. And we see an increase in both the numbers of delta FOSB positive cells and the intensity of their staining. And then in the cortex, if we stain for an excitatory synapse marker called VGLUT1, we see an increase in both the density and intensity of that signal. And this is actually really interesting because this is the first time that we're seeing effects in the brain outside of a neurogenic region. So this is in the cortex itself, and it would lead us to believe that there's something happening brain-wide. And again, we're actually injecting this IP, it's circulating systemically. It would make sense to us that it's having effects throughout the brain and actually throughout the body. So what is it that could be regulating or affecting both the angiogenesis and the neurogenesis in the brain? In order to get at this question, we collected blood from aged animals that had been either treated or not with GDF11, and we measured the amount of VEGF that was circulating in the serum, and we saw a significant increase after treatment. And if we culture primary BVECs, which are the brain vascular endothelial cells that line the capillaries in the brain and make up the blood-brain barrier, or the main component of the blood-brain barrier, if we grow those primary cells in vitro, we directly stimulate them with GDF11, we can see both an increase in GDF11 being secreted into the conditioned medium, so this is GDF11 and this is TGF-beta, and we can see increases in the expression of both VEGF itself and one of its receptors. And to us, this makes perfect sense because VEGF is well studied and typically thought of as a famous pro-angiogenic factor, but we've actually known for 20 years that it's also pro-neurogenic. So it's produced by both endothelial cells and by neural stem cells, and there's extensive autocrine and paracrine signaling back and forth between the two, and it actually acts to coordinate both angiogenesis and neurogenesis with each other. So I've shown you that both parabiosis and GDF11 can restore at least some aspects of a not degenerated, but an aged brain, some of the things that it's lost, like vasculature and neurogenesis. So there's something reversible about that process. And things like impaired blood flow and decreased neurogenesis are actually common across all aging phenotypes and in many different diseases of aging. And this has led us to this idea where we're asking, can we try and target diseases of aging in a more generic way as opposed to a disease-specific way? So instead of, say, going after Alzheimer's by trying to develop an anti-amyloid therapy, could you come up with a more generic strategy like improving blood flow to try and boost the cognitive capacity of whatever's left in the brain? And if that worked, it would be something completely new, but orthogonal and complementary to disease-specific therapies. And at least in these parabiosis experiments, there's something reversible about the degeneration that's happened. So maybe you could intervene in that way and actually restore some function to a brain that's already undergone degeneration. So where do we go from here? One obvious thing is to keep following these things up and move into disease models, try to dig down more deeply into the mechanism by which they might actually be working, and see if we can understand it better and intervene there. But another thing we could do that I just want to tell you about in the last couple of minutes is a project going on in the lab right now that we're really excited about, which is to use single-cell RNA sequencing analysis to try to identify all the changes that are happening in transcription across cell types in the brain during aging and parabiosis. And I'm going to tell you up front that I don't have any new results that I can show to you today because we don't have them yet, but this is something that we're excited about, and we think we're close to finding something interesting and having something that could be useful to the field in general. So what we've done is to take parabiotic pairs, either isochronic or heterochronic parabiotic pairs, dissect out the brain, dissociate the tissue into single cells, and then use single-cell RNA sequencing techniques to analyze the transcriptome of each of these cells. So the suspension is flowed through a microfluidic device where each cell is encapsulated in a droplet along with the reagents you need to make cDNA. And then we can collect that cDNA and sequence it with standard RNA sequencing techniques. And if you're not used to looking at this type of data, what you do is to then take each of those transcriptional profiles and use principal component analysis-based algorithms to cluster things together. So each dot here represents a cell, and cells that have more similar transcriptional profiles cluster together. And then you can retrospectively use the expression of known markers. For example, OLIG1, you can use the expression of known markers to try and assign identity to these different clusters that you've found. So OLIG1 is highly expressed in oligodendrocyte precursor cells and oligodendrocytes. And so by doing this, we've been able to analyze 45 brains, somewhere around 100,000 individual cells, representing about at least 16 different major cell types within the brain. And we now have this deep data set of gene expression changes that are happening during aging and parabiosis. Our bioinformatic analysis and our follow-up is underway, but we can already see changes in genes that have already been published to be age-associated. So APP, beta-2 microglobulin, IL-33, and a couple others. And we're hoping where this will go is that it's going to allow us to identify gene expression changes that are happening in the brain, and especially ones that happen in specific cellular subtypes. So something like the endothelial cells, which only make up a tiny fraction of the total mass of the brain, and in a lot of other experiments, the expression changes there get lost because it's swamped out by everything else. We might also be able to identify subpopulations, say excitatory versus inhibitory neurons. We might be able to identify pathways that are dysregulated, either in common across several cell types or specifically to one cell type versus another. We might be able to detect changes in the abundance of different cell types. And then we would really love to be able to identify novel druggable targets. For example, trophic factors that might be being secreted by the endothelial cells or other vascular cells into the CNS. That might be a completely new strategy to get at treating a neurodegenerative disease. So with that, I'd like to thank Lee Rubin, my mentor, Lita Katsamparty, who's now at the Pasteur Institute. She and Jaron Ozek are both postdocs in the lab who carried out all the SVZ and hippocampal work that I showed you. Methodios Zimirakis and Scott Lipnick are carrying out all the single-cell RNA sequencing analysis. Kathy Pfaff, Brittany Mayweather, and Rich Kraleski are the other members of our aging project team. And then our collaborators, Amy Wagers, Rich Lee, and Josh Levin at the Broad Institute. Thank you.
Thanks, Sean. It's a great thing about having young investigators speak. They stay on time and they are right on topic. This is great. A couple quick questions. Yes.
Hi, thanks for your talk. It looked like in some of the isochronic parabiotic pairs, old, that there was improvement in the brain vasculature as well. Is that true? And if so, do you have a hypothesis for why?
Sorry. Say it again. It looked like in some of the isochronic old, there was improvement in the brain vasculature.
Yeah. Earlier in your talk, was that true?
No, I don't think so. I think I only showed one picture of an isochronic old.
Okay, so when you put old, you don't see any improvement then?
No, just the act of parabiosis itself is not beneficial, at least in that context.
Thanks.
Okay, thanks, Sean.